Hello class, this is your lecture on plant tissues. Let's get started. Previously, we talked about plant cells. Cells are organized into tissues and tissues are organized into organs. After this tissues lecture, we're going to launch into all of the major organs that are found in plants. Our lecture outline is as follows. We're going to learn about all of these different types of tissues, including the meristematic tissues, which includes apical, lateral, and intercalary meristems. And then we will compare and contrast the simple versus complex tissues. First, a word about plant growth. Plants exhibit two growth strategies. One is primary growth, which is growth in length. This is often indeterminate in a lot of plants, meaning they can just keep growing longer and longer towards the sun all of their life. How does this occur? By cell division, specifically by a type of cell division called mitosis that occurs in both the stem and root apical meristems. We'll talk more about these meristems in the slides coming up. In some monocots, remember from our previous lecture, we compared the monocots to the dicots, and that monocot group, they often have a special meristem called the intercalary meristem. Now, other plants exhibit secondary growth. So while all plants do primary, only some do secondary, and this is growth in diameter. For example, things like trees with their woody trunks will often grow by secondary growth. This is by the activity of two different types of meristematic regions, including the vascular and cork cambium. It's also by some meristematic regions between some of the vascular bundles called the interfascicular cambium. So what are meristematic tissues? Well, meristems are permanent regions of active cell division. We can think of this as a group of cells that are capable of dividing and growing into any type of cell that the plant needs. You might have heard of things like stem cells in humans, where stem cells can be used to grow other types of cells and tissues for human surgery and stuff like that. Well, plants have their own type of stem cells that occur at these meristems. Now, the first type of meristem is very important. It's called an apical meristem, and it's associated with this primary growth. Therefore, it's found at the tips of growing roots and the tips of growing shoots. It increases the length of these organs as new cells are produced. Now, within this meristem, we find different regions, and these regions produce different types of cells. The first is the protoderm, which produces epidermis. The second is the ground meristem, which produces the primary plant body. And the third is the procambium, which produces the vascular tissue. Now in this picture, it shows us a micrograph, which is the fancy word for a microscopic picture of some tissues, from the leaf of a sundew. And a sundew is a carnivorous plant. It's able to eat insects because those insects stick to its leaves. Actually, it sticks to hairs on the leaves and then digests those insects as a way to get nitrogen. Well, if we look at this cross-section through the leaf, we can see all three of these regions that were found in that primary meristem as this leaf developed. We have the ground tissue, which is going to form the primary plant body. We have the dermal tissue, which is going to be the plant skin, and it includes amazing outgrowths like the sticky hairs that trap the insects. And finally, the vascular tissue, which transports water and dissolved substances through the plant. In this diagram, it shows us the relationship of the dermal, ground, and vascular tissue, and we can see that all three of these run through the plant in slightly different ways. We don't have to memorize those patterns at this point, but it is important just to know that dermal, ground, and vascular tissue are found in discrete areas. Our next type of meristematic tissue is the lateral meristems, and these produce the tissues that increase the diameter or the girth of roots and stems. We often refer to this as secondary growth. This is what we see in woody plants, such as gymnosperms like pine trees and dicots like oak trees. There are two cambium types responsible, the vascular cambium and the court cambium, and both of those allow for these trees to grow with lots and lots of diameter. In fact, some trees have grown so large that they grow right through fences, or they're big enough that a car could actually drive through it. It's phenomenal what this secondary growth can produce. The vascular cambium produces secondary tissues that function primarily in support and conduction, and the court cambium lies outside of the vascular cambium, but inside of the outer bark. And both of these are essential for allowing these trees to grow. Some monocots do have a modified type of secondary growth, but it's not the same as dicot secondary growth. It doesn't act by the same types of meristems. Instead, the monocots have a secondary meristem that produces parenchyma to the outside and secondary vascular bundles to the inside. This is how things like palms grow, coconut palms, for example. They're planted all over the Bay Area and definitely in Southern California. We often call these palm trees, but in reality they are not trees. They do not have the same woody tissues that we see in things like pines or oaks. But they do still produce a nice stem that looks like wood, but it's not wood. Now, monocots typically are instead going to have what's called an intercalary meristem, and these are special meristematic regions that develop along the stems, and it's from these nodes where we start to see growth. Often it's things like grasses that have these intercalary meristems, including a very robust grass, which is bamboo. Think about when you mow the lawn. When you mow the lawn, the grass isn't dead. It eventually grows back and you have to mow it again. That is because of these special types of meristems called intercalary meristems that are found in these monocots. So in review, how do plants grow? Well, they grow in a variety of ways. The meristems are going to be localized areas of 1, cell division, and 2, cell differentiation, which is to say, how do cells change into different types of cells? We've talked about apical meristems. We've talked about vascular and court cambiums. Another one is a marginal meristem. We didn't cover this one in great detail, but that's how things like leaves and petals grow into big flat objects. And then lastly, the monocots have the intercalary meristems. All dividing cells found in the meristems are undifferentiated. Later, they go on to develop into mature cells that have a wide array of shapes and sizes that are used later on. So now we're going to compare and contrast the different types of tissues. The meristems form cells, cells grow into tissues, and tissues grow into organs. And the first group is the simple tissues. The first type of simple tissue is parenchyma tissue. And parenchyma tissue is composed of parenchyma cells. It's a very simple type of tissue because, by definition, there's one cell type that makes up this tissue type. These cells look more or less like bubbles, where we see a huge vacuole in the middle and a primary cell wall. Two types of parenchyma tissue includes aerenchyma, which is parenchyma tissue with lots of interconnected air space, and chlorenchyma, which is parenchyma cells loaded with chloroplasts that form these chlorenchyma tissues. Our second type of simple tissue is cholenchyma. This is similar to parenchyma in that the cells are all living, but they are less uniform because the cell walls are differentially thickened. It's still a primary cell wall, but it has a thickness to it, and that is going to provide flexible support for growing organs. Very important for places like stems, where as the plant grows, it needs to be able to move a little bit without breaking. And the third type of tissue is sclerenchyma. This is cells with thick secondary walls that are usually impregnated with a biopolymer called lignin. All sclerenchyma cells are dead at functional maturity. This is because we often see these in places where the plant needs to be rigid. It needs to have a hollow tube that can move water, for example. Two different types of sclerenchyma include things like sclerids, which are referred to as stone cells. We often see these in stone fruits such as pears, and this gritty texture that we find in the fruit is the sclerid cells that provide support. Another common type of sclerenchyma is fibers. Fibers are often found in stems and leaves, providing additional support with its very thick cell walls. Again, all types of sclerenchyma have both primary and secondary cell walls, whereas parenchyma and cholenchyma only have primary cell walls. This image shows a nice comparison of cholenchyma versus parenchyma in a herbaceous plant stem. And we can see the cholenchyma has that irregularly thickened primary cell wall versus parenchyma just looks like bubbles, right? Because it just has a regular type of primary cell wall. Here we can see some fibers versus sclerids. The fibers with those very, very thick secondary cell walls, and the sclerids with those long pit canals that give support to this cell type in very soft tissues such as ripe pear fruits. So if simple tissues are made up of one single type of cell, complex tissues are made up of two or more types of cells. And it's not that this is made up of two single cells. It's made up typically of hundreds or thousands of cells, but it's two or more types of cells. Xylem is very important because this is how plants with vascular tissue move their water and minerals that are absorbed into that water by the roots. The xylem is made up of these different cell types. Vessels, which is made up of vessel elements. These are basically long tubes with open perforations at each end. Next is the tracheids. These are tapered at the ends with pits that allow water passage between the cells. And then finally we have the rays. The rays are typically made of parenchyma, and this is going to be for lateral conduction from the center of the organ to the outside. For example, from the center of a stem to the outside of a stem. These vessels and these tracheids often appear side by side in those vascular plants. In tracheids, these pits tend to form whenever they come in contact with each other. Here we can see the tracheids versus the vessels side by side. You can see that the tracheids have pits on the ends but not big openings or perforations, versus the vessels have big openings. The general term when we're referring to these types of xylem parts is tracheary elements. Water moves passively by transpiration through these tracheary elements, and they are very important for plants and their metabolism. Our second type of complex tissue is phloem. Phloem moves the food materials, which is usually sugars that are all made by photosynthesis. Phloem tissue is made up of several different cell types, including sieve tube elements. They're also referred to as sieve tube members, which are large cylindrical tubes. Sieve plates, which are the porous regions on the ends of the tubes, and companion cells, which are narrow, tapered, and nucleated, meaning they have a nucleus. So the sieve tubes are similar to the vessels in that they are dead at functional maturity. They have a thick secondary cell wall. But companion cells are alive at maturity because they have a nucleus. The companion cells have to direct the loading and unloading of sugars into these phloem tubes. Our third type of complex tissue is the epidermis, and this is the outermost layer of cells that are found on plants. We often see an epidermis on the roots, the stems, and the leaves. It's usually 1 cell thick, but sometimes more than that. And this epidermis almost always secretes cutin, which is a fatty substance that covers the outside of the outer cell walls. On leaves, this usually forms a cuticle, which is very important for providing sunblock and water preservation in those organs. Root epidermal cells often produce root hairs to increase the surface to volume ratio of these organs, and leaves usually have special openings or holes in the leaf called the stomata. This opening, which is the stomata, is formed by a pair of guard cells that open and close. When we look at the surface of the epidermis, sometimes we see little hairs, and the fancy word for these hairs are trichomes. These would be considered to be epidermal outgrowths. Our fourth complex tissue is periderm. Periderm is what makes the outer bark in woody plants. Periderm essentially replaces the epidermis. As the periderm forms, the epidermis falls off, and it never comes back because now the plant is a woody plant with secondary growth. The periderm is primarily composed of cork cells. That is to say, the cytoplasm of these cork cells are very important in that they secrete suberin, which is a waxy substance that provides very important waterproofing for this bark layer. Some parts of the cork cambium form a loosely arranged pocket of parenchyma cells that actually protrude through the surface of the periderm. This is referred to as a lenticel. You can think of it as an opening or a pore in the bark that allows the woody tissues to breathe. Here is a diagram that shows us the relationship of all of the parts of the periderm to the epidermis and to the innermost layers. The main thing to get out of this diagram is that the cork cambium produces the cork cells to the outside and the phelloderm to the inside. Another name for the cork cambium is the phellogen, which is spelled Phellogen. This phellogen is a word we'll see in some textbooks. The cork cells, remember, have suberin in their cell walls to provide waterproofing, and the phelloderm is similar to parenchyma. Our fifth and final complex tissue that we will learn in this class is the secretory tissues, which of course is made up of secretory cells. Cells may function individually or as part of a secretory tissue, as part of this complex region. And this is how things such as flower nectars, citrus oils, glandular hairs that produce mucus, latex, and resins are produced by plants, just to name a few. We've covered all of the tissues that are important to allowing plants to grow. After listening to this lecture, you should understand all of the meristematic tissues, the simple tissues, and the complex tissues. That's it for this lecture. Please email me anytime you have a question, and thank you for listening.